Okay, so up until now we've been talking about gram-positive bacteria with the staphylococci and the streptococci. Gram-positive bacteria having this thick peptidoglycan cell wall, which retains crystal violet during gram staining, giving it the purple colour. And then now we're going to move on to some gram-negative bacteria with a thin peptidoglycan cell wall and an outer membrane that picks up the crystal violet but gets washed away with alcohol during the gram staining process, which gets rid of the purple colour, and then the counter stain of safranin means that the gram-negative bacteria are going to be kind of pinkish red. So gram-negative bacteria are the largest group of human pathogens. Their cell walls contain lipid A, which stimulates fever, vasodilation, and inflammation, as well as shock and disseminated intravascular coagulation. Basically, it causes blood clots throughout the body. There are approximately 30 genera of gram-negative human pathogens. A few examples are Neisseria, which is a kind of cocci, with the kind of spherical shape, the same as Staphylococcus and Streptococcus, except it's gram-negative. The Enterobacteriaceae and Pasteurellacea, which are facultative anaerobes comprising 13 genera, which we're going to cover in quite a bit of depth in this playlist, but probably not in this video. The Pseudomonas, which is a kind of aerobic bacilli, and the Bacteroides, which is a kind of anaerobic bacilli. Bacilli just meaning it has this kind of rod shape rather than the round spherical shape of the cocci. So the Enterobacteriaceae, or enteric bacteria, are part of the human intestinal microbiota, with some being deliberate pathogens and others being opportunistic pathogens. Deliberate pathogens meaning that their life cycle requires a human host, so they're deliberately trying to infect you. Meanwhile, opportunistic pathogens are pretty much always present on us, but they are only able to cause an infection if conditions are right, like if they're moved from where they usually live to another place, or if the barrier of the skin is broken. The Enterobacteriaceae account for most nosocomial infections, so those picked up at the hospital. Sorry, it's quite early in the morning. The enteric bacteria have a peritrichous flagella, if motile, a prominent capsule with loose slime to help them evade the immune system, and they all reduce nitrate to nitrite and ferment glucose anaerobically, but they tend to grow better aerobically, which is why they're facultative anaerobes and not just anaerobes or obligate anaerobes. So they're basically aerobes that are able to also ferment to produce energy that way. They are all oxidase negative, so they don't turn purple in an oxidase test. Enteric bacteria tend to be coccobacilli or bacilli, so that rod shape shown on the right here. And they're typically tested for via blood agar hemolysis, which we've covered in a previous video, or something called a McConkey agar test. A McConkey agar test, basically the agar contains bile salts and crystal violet that inhibit gram-positive bacteria. So gram-positive bacteria are not able to grow in that petri dish with the agar. So any bacteria that are growing in that petri dish are going to definitely be gram-negative, because only gram-negative bacteria can withstand the crystal violet and the bile salts that would normally inhibit gram-positive bacteria. The outer lipopolysaccharide membrane of enteric bacteria, so that would be this, if you look at this image up here, the gram-negative one on the left, you can see the peptidoglycan cell wall in purple in the middle, and then look to the very top, you've got the outer membrane. That is called the outer lipopolysaccharide membrane, and it has three parts. The core polysaccharide, shared by all enteric bacteria. The O polysaccharide, which has various antigenic differences between different species of enteric bacteria. And lipid A. Other antigenic components include protein and polysaccharide capsular K antigens, and flagellar proteins called H antigens. So K antigens just mean they're coming from proteins and polysaccharides. H antigens coming from the proteins from the flagellum when it kind of breaks apart, and it can be picked up by the immune system. Serological identification of each of these 5 antigens is used to distinguish between strains and species of enteric bacteria. Enteric bacteria virulence factors, molecules that aid in infection, include lipid A, the capsule, the fimbriae, exotoxins, iron-binding compounds, hemolysins, type 3 secretion system mechanisms, which are the things responsible for injecting the virulence factors. So virulence factors, that is things that help the bacteria infect you and reproduce. So the family of Enterobacteriaceae, we have a few different types. Opportunistic coliform bacteria, which produce spores, and they're typically found in water, soil, and the human intestinal tract. It includes these species shown here. The first one you should be familiar with, Escherichia coli, and we're going to go deeper into it probably in one of the next videos. We have opportunistic non-coliform bacteria, which cannot ferment glucose, so I don't think I just mentioned it then. Coliform bacteria just means that they can ferment lactose, so all these are coliform. Non-coliform bacteria cannot ferment lactose, and then they're shown there. And then we have truly pathogenic Enterobacteriaceae like Salmonella, Shigella, and Yersinia, which you should remember because we're going to go over that probably in the next video. So, a quick overview of Escherichia coli, E. coli. It's a kind of opportunistic coliform bacteria. It's the most common and an important coliform bacteria with numerous oxygen, hydrogen, OH, and K antigens to identify different strains. Remember, we use these OH and K antigens for serological identification of gram-negative bacterial species. Virulence plasmids can also be transferred between individual members of E. coli, so that can transfer stuff like antibiotic resistance. E. coli is responsible for urinary tract infection, neonatal meningitis, which is inflammation of the meninges in unborn babies, gastroenteritis, which is caused by the bodily response of vomiting and diarrhea to enterotoxin, with rapid loss of electrolytes, which is kind of a lot similar to just basic diarrhea. It's treated the same with water and water containing electrolytes and sugar. And hemorrhagic colitis. As you can see, most intestinal infections and UTIs that will be identifiable through bodily excretions. The E. coli strain responsible for hemorrhagic colitis produces large amounts of cytotoxin called virotoxin, which kills body cells. E. coli infections are treated with various antimicrobials and can be prevented by avoiding fecal contamination of food and water, plus good personal hygiene.